for us. I realized, getting ready for the talk today, I started coming here in 2010. It's been a while. I think Boris was actually here at that time. I think this is my 5th time back. KITP's had a huge influence on me. I think when I first came here, there was a session on, maybe the first ever, on evolutionary cell biology, and I was just brought in as the token evolutionary biologist, I'm sure, but it really transformed my life. And now I'm working on cell biology things, and I'm convinced there's a lot of room for progress there, including with biophysicists. So for the physicists in the audience, please interrupt if you need some vocabulary checks. There's a couple of physicists at our program, so they will presumably know where I'm not defining things well enough. And as you'll see, there's a couple of tricky spots here, but there's also some real parallels to what we're doing and some of the basic mathematical procedures that you guys use in like statistical mechanics and things. Our program, I think, is called Population and Quantitative Genetics of Adaptation. And I might not be the best spokesperson because actually a lot of my work is focused on non-adaptive aspects of evolution. I don't believe you can explain everything in evolution just based on natural selection. So understanding natural selection is one of our great achievements in the biological sciences, but it's one of our big problems, I think, in evolutionary biology, because for most biologists, most scientists in general, it's thought that there's nothing more to evolution than natural selection. You go and compare it to organisms, they're different, and you make up some stories for why they're different, and you try to test it, and sometimes it doesn't work, and you try the next year to test it again, it doesn't work, and next thing you know, you've spent your entire career on this, and you know, you collapse in a heap on the floor, I guess. So let me just start out with what I mean by this. I've tried to put up some stuff on the board to get things going, because this is my first use of the chalkboard. I've been using these Zoom boards. I don't know if any of you guys know how to do that. If you do, please teach me, because I'm not very good at it. So I figured I'd put some stuff up here to start with. So the starting point here is what I like to call the population genetic environment. So evolution doesn't just operate in a vacuum. It depends on various features of the individuals in the population that you're studying. And I like to think of there being 3 dimensions to the population genetic environment. So one is mutation. Nobody's figured out how to stop mutation from occurring, and I'll give you some numbers on that in a second. So mutation is the ultimate source of variation upon which natural selection operates. So we're talking about mutation at the DNA level. Pretty sure everybody knows what DNA is, correct? Comes on these long strings we call chromosomes. And I'll get into that in more detail in a second. Recombination occurs in a number of organisms. You probably have heard of crossing over. Not all recombination is crossing over. In fact, only a small fraction is what we classically think of crossing over, where you exchange flanking markers between dad and mom's chromosome. But it reassorts variation within and among chromosomes in organisms, so that can be quite important. It can be really good for evolution in some cases because it can bring 2 independent mutations together that need to be brought together to make an adaptation. On the other hand, it can rip a good thing apart, so it's sort of a two-way street. And then finally, there's this thing we call random genetic drift, and I've intentionally underlined the word drift here, because in population genetics, we use the word drift in different ways than you guys in physics use the word drift in your diffusion equations. I believe you generally think of drift, and it sort of makes sense, as a directional force. When we talk about drift in population genetics, that's our background stochastic variation. That's the non-directional force for us. Otherwise, it's more or less the same kind of diffusion theory we use, as we'll see in a second. But I just want to make sure when I'll be abbreviating random genetic drift to just the word drift. And so keep in mind, that's the noise in the evolutionary process. It's not the directional force.
What's the difference between mutation and drift?
Well, mutation we think of as a reasonably deterministic thing. I'll give you some rates in a minute. So you've got a nucleotide A, it changes to a G. And once it's inherited, it's permanently a G forevermore. Drift, as we'll see in a second, is random fluctuation. Suppose you have what we call a polymorphism. It's a site that some individuals in the population have an A and some have a G. So there's a frequency of A and G. And drift refers to the random fluctuations in the allele frequency of A from generation to generation. Once it hits 0, you're stuck. The only way back in is by mutation, or if you hit 1, you're stuck as well. But drift is referring to changes in gene frequencies in populations over time.
So what is the quantity that drifts and stuff? Is it a letter? Is it a number of genes, something like this?
Yeah, I'll get to that in just a second. And in fact, that's coming up here. And this actually will connect two different approaches we're taking here in this program. So I'll be using this word, or this abbreviation NE. It stands for effective population size. So you can sort of think of drift as sort of the coin flipping problem, right? You have a coin and call heads, you know, the big A allele and tails the little A allele. And if you flip the coin 1 billion times and it's a fair coin, then you'll be very close to 50% heads. But if you flip the coin once, well, it's just going to be a head or a tail. It's a major flip in the expectation realization, right? Or if you flip it 10 times, you're probably not going to typically get a 5 heads. And that's essentially what's going on. I think for practical purposes today, we can think of that's what's going on. Every time populations reproduce from one generation to the next. So drift is occurring in transmission of allele frequencies from one generation to the next.
Whereas mutations is defined for a single, I mean, it's like a cosmic ray.
Yeah, mutations occurs in single individuals at a time, and they're, as we'll see, they're very rare events, so it's unlikely at 2 individuals. Well, if we're talking about huge microbial populations, 2 individuals could get hit at the same spot. But for things like us big crunchy things, animals and land plants, it's unlikely that the same nucleotide will get hit in 2 individuals. Okay, so this thing will become important that we call NE, the effective population size, and there's, some people don't like the use of this term. I like it quite a bit, as long as you define what you mean by the term. The problem is, in population genetic theory, there's lots of kinds of effective population sizes. What we'll be thinking about it today, like, is it's the population size that has to, all populations are non-ideal in form. And so we can think today about the effective population sizes. How is all the complexities about biology, sex ratio, environmental variation, variation in family sizes, how is this influencing the variance and change of allele frequency from one generation to the next? And as NE gets small, that change is high. And everybody has an effective population size, much, much smaller than the absolute number of individuals in populations. There's actually a way to make it bigger that I won't go into today, but you know, I think we can double it by doing certain breeding schemes. I'll just leave it at that. That's not going to happen in natural populations. But I'll give you some numbers now. So,
The effective population size depends on the time you're considering for the process.
well, I don't know if I want to get into this, but it could be flipped over and turned into a time. So if you think about a population and a gene, there's really a genealogy of that gene and everybody in the population, and eventually it will do what we call coalesce to a common ancestral gene sometime in the past. So like mitochondrial Eve or Y chromosome Adam, if you like. Under an ideal situation in which there's no selection, it takes about 4 NE generations if you're a diploid species, which we are. It takes about 4 NE generations to go back to a gene genealogy in a population. So it actually has a dimension of time because it's influenced the rate of diffusion. That's essentially what's going on. Okay, so what about this thing called NE? We've been doing a lot of work over the past few years on mutation. I'll talk about a bit. I'm not going to tell you where we get these estimates of NE in great detail, but it comes from measures of variation in natural populations at certain sites we believe are close to neutral, in which case the only other thing influencing the standing variation is the mutation rate. And we know the mutation rate for about 40 or 50 organisms now. We've spent, I don't know how many millions of dollars doing this, but we know it. So there's a bunch of organisms we have a pretty good estimate of this. And it turns out if you plot NE versus the adult mass of an organism, this would be a log scale here on both axes, you get a regression with a slope of about minus 0.2. And so this is as bad as it gets. So some mammals, including us, have a long-term effective population size of about 10,000 individuals. Now we're clearly beyond that today, but historically, because everything here today originated, thousands of years ago, this is what we are historically. And this is not far from what you see in a lot of vertebrates. At the upper end of the scale here, it doesn't get any better than that. Even bacteria that might have an absolute number of cells in the population of 10 to the 20th, the highest NE we've been able to measure in anything is just short of 10 to the 9th. So that just gives you an idea. And I'll explain more about why this happens in a minute, but part of it, I think almost all of it in microbes, is due to the fact that all our nucleotides are tied together on chromosomes, and that causes what we call selective interference. It sort of causes the eyes of natural selection to glaze over, so to speak. Okay, we okay to this point?
What is S is the slope?
Yeah, so, well, S is something I'm going to define right now. So what I've talked about up to now is what I call the population genetic environment. There's one other thing so I've just told you about how NE scales with the size of an organism. The bigger you are, on average, and I'm talking about things across the whole tree of life here now.
So this simple scaling presumably has some kind of geometrical...
Well, I can't give you a mechanistic reason why this falls at minus 0.2. Other than the fact that in different organisms of different sizes, the total mass in the whole species is roughly constant. Roughly. I mean, I'm talking orders of magnitude here. So in general, the bigger things get, the smaller the number of individuals. But this doesn't scale at all with N. So as I said, a bacterial population might be 10 to the 20th, in some cases even larger. And, there are mammals down around 10 to the 5th or so. But it's not a linear scaling. Okay, S. So now we'll get into selection, and that will tie back to here. So you can think of 1 over 2NE as like the number of times your coin is going to be flipped each generation. And so, just think of a standard deviation for a binomial distribution, right? You've got a number, the square root of some number in the denominator. So this is governing the noise in the evolutionary process. S is what we call a selection coefficient. So suppose that you have some nucleotide, we'll say it's A. We're only looking at one strand here. DNA is double-stranded. We'll just focus on one strand. And then maybe other individuals have a G in the population. We're extremely fortunate, gift of the gods or something, but the mutation rate's low enough. So almost never do you go out into the wild and sample DNA from different individuals. Usually, there's no variation at all. 90% of the sites, you'll find no variation among individuals. Sometimes you'll find 2. Some guys will have a G and some guys will have an A. You almost never find 3 in a sample of a few hundred in the natural population. So I'm keeping things simple, but there's a basis for keeping it this simple. So what you can think of here is, in terms of selection, is we'll just scale everything. So you have a certain fitness if you have an A. And we're going to keep things simple here, so you're no longer diploid, you're haploids. Hopefully nobody in the room is actually a haploid, but one never knows. And that just keeps the math simple. And it works for diploids too, it's just a little twist, so we won't worry. They're just trying to keep things simple. But suppose you're born with an A. Lots of organisms, by the way, are haploid. All bacteria are genetically haploid, and lots of unicellular eukaryotes are haploid. But suppose you have a G, and maybe having a G versus an A boosts your fitness up by some small amount, S, where S is some number much smaller than 1, and often much orders of magnitude smaller than 1. That would be a beneficial mutation, and if this is a minus, then it's a deleterious mutation. And so selection would be favoring A's. That's what natural selection does. It favors alleles with higher fitnesses associated with them. Now, there's everything's a matter of relative power of different evolutionary forces in evolution. So the power of random genetic drift in a haploid is 1 over the effective population size. So that's the noise in the evolutionary process. And we can think of S as being sort of the directional force operating to increase or decrease the allele frequency. And the way it works out using diffusion theory, or you can do it in other ways, but if S is less than 1 over NE, selection loses because the noise, it's constantly pushing you along in one direction, but the deterministic force of selection is being overwhelmed by the background noise, and the noise wins. So we call this effective neutrality. For any mutation that has an effect less than 1 over NE, selection can't touch them. That's why this becomes important. Because if you live down here and NE is 10 to the 4th, that means that any absolute value less than 10 to the minus 4? Just the inverse of NE. That means it's invisible to the eyes of natural selection. So that means if you're you're some animal or land plant, natural selection can't use any mutations that have benefits that are less than 10 to the minus 4. And it also can't remove deleterious mutations if the effects are less than 10 to the minus 4. They'll just accumulate passively in a neutral fashion. And so this is an important benchmark. On the other hand, if you're up here, you can utilize a mutation that has a benefit as small as 10 to the minus 9 and eradicate one that has a detriment on your fitness as small as 10 to the minus 9. So that means there's about 5 orders of magnitude differences in S. These guys can really fine-tune evolution, take advantage of mutations of small effect, where evolution must be more coarse-grained out here. That's the sort of the cartoon version of it. And this is important because we know some things about what S should be, and there are certain cases, just to make a point in a simple way. Suppose you're going to add 1 nucleotide base pair to a genome. We know that you require about 50 ATPs. This is our energy currency in biology. It takes about 50 ATPs to make a nucleotide, and nucleotides come in pairs in DNA. So if you want to add 1 base pair, that's 100 ATPs it's going to cost you. It's a small number because the cost of building a cell about the size of an E. coli cell is about 2 billion ATPs. That's how many ATPs it costs to build an E. coli cell. And so you can think of the selective disadvantage of adding 1 nucleotide to an E. coli genome. Let's just round this off to a 1. Right, that's what you do in physics all the time, right? We talk in orders of magnitude. And the numerator is 100. And so that's a 10 to the minus 7, I guess. Sorry, I'm off by a factor here. This should be a 10. And so this is a 10 minus 8. So that means you could add 1 nucleotide to a bacterial genome in a natural population of bacteria that has an effective size of 1 billion. And natural selection could feel it. There's no way it could feel it here, and that means that small insertions just passively expand in genomes of us big things with small n. That's the idea. Just to give you an idea. I mean, usually we can't calculate S directly, but this is an example. This nucleotide insertion, I'm assuming here is not knocking out a gene or anything else. It's just some nucleotide in intergenic region. The only thing it's doing is costing you the ATPs that it took to make. So that's selection, effective neutrality. And now I think I'll move on to thinking about the analogy about the two ways physicists think about things in the physical world and the parallelism between the topics we're exploring in the population and quantitative genetics realm here. So physicists like to think of microscopic behavior, say all the molecules in a gas or something, and you like to think about you have these things called the Boltzmann equation and so on that explain all the possible microscopic states of the billions of molecules in your substance. And sometimes you'd rather think of, I mean, that's kind of daunting, so sometimes you'd rather think about just the macroscopic feature of your gas, the pressure in your gas volume and so on, the emergent properties of having all those little molecules jiggle around. Well, we have a microscopic and macroscopic view of evolution in population versus quantitative genetics, and they're both completely compatible. This field, quantitative genetics, as we'll see, ignores all the microscopic properties. Not always, but I'll get into this in a second. But if you're thinking about population genetics, you're usually thinking about the change of frequency of some allele, like this big A allele, over time. You're thinking about changes in allele frequency. Sometimes you might get adventuresome and think about the joint changes of two allele frequencies at two loci that might be linked together, maybe not. 3, we prefer not to ever think about. What was the saying the other day, 1, 2, 3, infinity? That's sort of the break point. But just to give you an idea, the way to think about this, and you'll see, I hope the physicists will see a connection with some famous equations in physics. So suppose we have two alleles here. We'll call them the plus and the minus. That could be our A and our G. The plus is good, the minus is bad. And they're mutating, and they're capable of, an A can mutate to a G just as a G can mutate back to an A. So these are our mutation rates in both directions. And beta then, the ratio of those rates is what I call the mutation bias. So if mutation from A to G is the same as G to A, that ratio is just 1. But, maybe A is good and G, C, and T are all bad. Then there'd be, even if there's nothing going on at the molecular level, and the bias would be 1 to 3, because there's 3 ways to make a bad, but only 1 way back to a good. Make sense? So that's mutation. And then what you can think about is, well, what happens now if there's selection going on? So let's make G an advantageous allele. There's selection going on. That's the selection coefficient S that we just saw. And then there was this thing, NE, the effective population size. And as I said, the ratio needs to be greater than 1 for selection to be very efficient. And so if we just rearrange this, you see that NE and S come out naturally together. NES is the ratio of the strength of selection to the strength of drift. That's the way to think about it. Well, in a simple world where the population size isn't too great, you can imagine a population starting with a G. And then a long time goes on and somebody with a G mutates to an A. And usually that A, it's very rare, so it's by chance going to be lost most of the time, but occasionally it will get even though it's slightly deleterious, it might eventually go to a frequency, from a frequency of, initial frequency is going to be 1 over the number of individuals. And if it somehow wanders around and goes to a frequency of 1, we call that fixation of the A allele. And now you're sitting in the state A for a long period of time. There's this long waiting time. And lo and behold, A mutates to G in somebody, and that wanders to fixation. Actually, G is advantageous, and it goes to fixation. Okay, so there's this other term here that turns out to be quite important, e to the 2NES. That's the ratio of the probability of fixation of a beneficial allele, starting at with one copy in the population. This is the ratio of the probability of the beneficial versus the probability of fixation going in the other deleterious direction. So this is a really elegant, simple result because the numerator and the denominator, those aren't so simple, but a whole bunch of stuff cancels out. And so this becomes important. You can think about this. Beta is the mutation pressure in the upward direction. Well, this is the selection pressure in the upward direction. And the product is just the net pressure towards the beneficial allele due to mutation and selection. It's as simple as that. And then in this simple world where everything's sequential, like I just said, you're in one state, pure state, sitting around waiting, and then finally you go to the other state. You can then ask, well, what's the probability? Let's just say we're following our population every million years. We're sequencing it and finding out what state it's in. And in the long run, you'll get a steady state distribution if we keep S and E and the mutation rates constant. And there'll be some probability of being in the beneficial state, P squiggle, and that's equal to this quantity here. And I think that form of that quantity might look familiar to some physicists, right? That's something like what you call the Boltzmann distribution.
You haven't defined beta.
Beta is the ratio of mutation rates in both directions. So maybe you guys, do you call it the chemical potential, something like that? So that's the pressure in the A direction just based on mutation alone. And then selection, if it's effective, will push it along even further. Of course, it could be deleterious, in which case this would be e to the minus 2NES, and then you'd have an opposition between mutation pressure and selection. But you know, this is really the same as what you guys call the Boltzmann equation, right? And for alternative molecular states. And the denominator is, I believe, what you call the partition function, yeah. Okay? So that's simplest population genetic scenario. It gets a little more complicated if population sizes get larger because no longer are you always in one pure state or the other. In fact, if the population is large enough, it will always have the beneficial allele and the rare one will just be kept there at low frequency. And people have worked that out. You do some interesting math that involves like ratios of confluent hypergeometric functions that we don't want to talk about. But the bottom line is if you plot this and you're thinking about allele frequency or the beneficial allele, at small n, you're way down here. That's the expected frequency. And NE so small, this goes away, and it's just beta over 1 plus beta. And then as you get to larger and larger NE, selection dominates, and you go up to almost 1 minus the probability of having a deleterious allele. And the pivot point here is pretty close to this. So to the left of this NE, drift and mutation dominates, and to the right, selection dominates. So that's the simplest world. I'm glossing over a lot of details, but I think that sort of reasonably accurately conveys things for I'm interested in long-term divergence among populations of organisms of very, very different types. And the beauty of this, you can see here, is that just depending on the population size, not changing selection, not changing mutation, the mean phenotype is going to change. You're going to get a gradient in mean phenotypes, even keeping selection and mutation constant. Now let's jump over to here, the macroscopic world, where things obviously will potentially get more complicated, although we like to develop models that really simplify it, and no longer taking a view of the allele frequency At one extreme, we can assume there's an infinite number of alleles, if you like. But we just focus on the means and the variances of some trait. So now you have a trait, like height or something, that's just the sum of effects from all the different genetic loci put together. And so now we're measuring some trait that you could all do with a yardstick or cell phone, whatever we do these days. And we know Z for everybody. And so now we think about the fitness function as it's no longer you're just this type and we've got a fitness 1, and this type you've got a fitness 1 plus S. Now you've got a whole range of phenotypes like height or something, you know, number of bristles on the left leg of the blue dicky whistle or something like that. And now we can measure what we call the fitness function. And so now we're asking, given you have a certain value of Z, what is your fitness? And this can take any number of different forms. We like to work with a thing called the exponential fitness function if most alleles are deleterious, and Z is the number of deleterious alleles, then your fitness would decline exponentially with the number of deleterious alleles. Some people like to think about, a lot of people like to think about Gaussian selection, where there's some optimum phenotype in the population, and then the width of that is a measure of the strength of selection. So the greater the width, the flatter that curve becomes, the weaker selection becomes. You might have a MESA function for like biochemical properties. And so that just levels off. It's asymptotically approaching some high value. So those are fitness functions. And then we like to think about if mutation is bringing in new variation each generation, and so on, spreading things out, then selection's doing something, and all this works together, more or less like this back here, but on a grander scale, and you will get an equilibrium mean phenotype distribution. Okay? So here we're really hiding all the genetic details. They're there, but we're trying to collapse it all into these microscopic properties, just like you do in physics many times. Classical physics.
There's a way where the microscopic traits can be expressed in terms of microscopic.
Yeah, yeah.
What about here you don't have that.
Oh no, you can do absolutely that, but the bookkeeping gets rather horrendous. So you know we're talking about sometimes thousands of loci. So generally we don't go down. That's why I'm saying it's sort of 1, 2, maybe 3. And then we just jump to assuming there's an effectively infinite number. But people have spent a lot of time actually deriving the link between the microscopic features of the system and macroscopic. But you know, those aren't fun papers to read. But the main point is it's been done. Not in every detail, of course. Okay, so there we are. And here's the problem, the additional problem that arises. Our nucleotides are present together on chromosomes. When I was talking here, was as though nothing else in the genome mattered. Their selection was paying attention to this one nucleotide in the genome. Everything works out nicely then. But in reality, our nucleotides are linked together on chromosomes, and so something like yeast, the average chromosome length is about 1 million bases. And us, what is it, 3,000 divided by, rounded off to 30, 100 million bases, something like that. I might be off by a 0 or something, not very good at math. But the point here is you may have a lot of these sites now that are all under selection simultaneously. Like, you know, selection might be favoring in some optimal wing length or something. And let's just keep things simple. There's a good allele and a bad allele. And suppose there's just 6 sites here that are important. And natural selection's favoring 3 pluses and 3 minuses. Well, there's multiple ways to get those 3 pluses. Right? So here, there's 3 pluses, and here there's 3. They're completely different sites. But as far as natural selection is concerned in this simple additive world, they're the same. Okay, now, if one of these came along compared to the other one, natural selection wouldn't be able to distinguish them, and one would be just drift to fixation over the other, unless there's recombination between the sites, because then you could sort of shuffle things, and it doesn't operate at this rate at the single nucleotide level, but you can imagine if you had the right kind of recombination, you could make an advantageous chromosomal type with pluses in all the sites. Okay, but I'm talking today sort of about what we think about as linkage groups. They're small enough stretches of DNA so that by the time you get back to this thing I called the coalescent, there's no recombination, no appreciable probability of recombination in there. So that's where we are on that. And then, let's see, I think I might have drawn some stuff up here relevant to this. Yeah, so, well, I meant to mention this recombination rate thing before. So we know a lot about recombination rates in organisms. And this is one of the closest things that, I'd be willing to call a law in biology, is the average amount of recombination per physical distance on a chromosome in different organisms. And we know this from lots and hundreds of what are called genetic maps. And if you plot the recombination rate, the crossover rate per base, the average crossover rate per base across the whole genome, as a function of the average chromosome length, you get a slope of -1. We can explain the average recombination rate to an accuracy of about 99%, just knowing the average chromosome length. And there's a simple reason. Well, there's a reason for this. We don't know where it came from really, but there's roughly, when we undergo meiosis, there's roughly one crossover event per chromosome arm. So if you double the length of a chromosome, you've reduced the amount of recombination per base by 50%. And so that's what you get here, and these numbers can be really low. Yeast, I don't remember the exact number. Yeast is as good as it gets. I think it's around
110 per meiosis.
110 per meiosis? Okay, then we have to divide by 10 million. So 10 to the minus 4 maybe? I might be off a little bit. And then us guys, we're way down here around 10 to the minus 9. They're roughly on the order of the same magnitude of the mutation rate per base, plus or minus a order of magnitude. Okay, so where are we going here? I think, so I guess there's 3 more things that I want to mention. So if we follow this model here, we have a fitness function. It tells us how fit individuals are with different values of your trait. And then let's suppose these guys are all mutating with the same scheme I had before. For every site, a plus is going to a minus at rate U10 and vice versa. So we have the same mutation scheme, but things are bound together in bigger ways. Now, it turns out that for a lot of reasonably defined models, you can predict what the mean phenotype will be as a function of the population size. We actually just did that for this. There's only one nucleotide here, so the mean phenotype is directly proportional to the frequency. That's just the average time you're in A versus a G. But you can play the similar game with this kind of analysis. And the way it works out is that, so what we'd be after is the probability of an individual in the population having some value Z. That might be your height or something. And there's going to be some distribution in the population. Another thing I'm very interested in is the binding strength between 2 components of a molecular structure. And Z there might be the number of hydrogen bonds, from 0, in which case your monomer is floating off by yourself, to a 10 or something, in which case you're really stuck together. You can think about this in a lot of different ways, but there's some value Z. And for a lot of models, it turns out that if we just think of a simple linear case where every additional value of z advances or reduces your fitness by s, you end up with a Poisson distribution where the distribution of p over z, of p of z, is just Poisson in the population. And it's just a function of one. The good news is it's a function of one parameter. The bad news is that one parameter has mutation, drift, and selection all in it at the same time. And if any is small, this goes away, and it's just beta. That's just where you would be down here under. It would be it would be a binomial with roughly binomial. It's Poisson with the parameter beta. And so you would have, I like to think of this as the null expectation. It's where you would be if there was no selection whatsoever. Okay? And the way you would think about this is if you're studying this population with small n-e, or any population, you're just censusing it every 10, 100 generations or so, and you're building up a probability that the population is in all these different states over time. And it'll wander. It won't stay at exactly the same mean phenotype because the stochasticity of mutation, in this case drift, allowing you to move around. And then, as you add in selection, this is a quite neat trick, is all we're doing is scaling this neutral expectation by e to the 2 NES. And it just transforms the neutral distribution into this other distribution. So as NE gets larger and larger and larger, it transforms this thing that's roughly Gaussian. It moves it over, moves it over. We're not allowed to go beyond that point. That's the biophysics barrier or something, right? That's some upper limit to, say, a chemical reaction rate or something. But you will move from the left to the right. And the couple important points here to make is biologically, since a lot of us do comparative biology and like to go out and study organisms and what we think are different environments, at least what we think the organisms thinks are different environments, and then we study mean phenotypes, and then we look for correlations, and then try to explain the selective differences. Well, this kind of result, and I've oversimplified a few things, but there are things that, some statements that come out of this, I think, that are very general, and that is that so nothing has happened here in wandering from the left to the right, except I've increased the population size. And at any one population size, you have this distribution. So even with constant drift, selection, and mutation, the population won't have the same mean phenotype from generation to generation. It'll wander around within some confines that are determined by these processes here. But then, as we...
Does this include changing species, let's say? Like, can I go from an amoeba to a fish? Something like this?
Well, not in my lab, but that would be the idea. I'll give you a final example that I think will get back to your point. I'm thinking, big scale across the whole tree of life, from microbes up to us vertebrates. The point here is that if you have a trait that's under constant selection, this might be true with like the enzymes in the Krebs cycle or something that are doing the same thing in every organism and always have been. DNA polymerase, its job in everybody is to copy DNA. So if you have a trait that you could measure in all organisms, the prediction would be that in organisms with larger NE, smaller size, these distributions would move to the right. And even with a constant size, you'll wander around a bit. But this predicts a gradient in mean phenotypes without any change in selection across species. It's just due to the change in the power of random genetic drift. So let me, to me, that's a pretty strong statement, but you really don't need to do the math in any great detail to understand the principles, I think, underlying this. This has to happen because mutations arise stochastically and drift is a randomizing process. So let me tie this all together with a couple of examples back to biology now. And these are patterns of variation in a trait across the tree of life. So one is illustrated here. So before let me just forget what I said before. Don't forget what I said before, but let me just move on. I want to make a summary of a lot of observations we've made over the past, 15 years. We've been measuring mutation rates in about 50 organisms, and it's a lot of work. We start with like 50 replicate copies of a genome. We bottleneck them every day till the technician gives out, and then we sequence the whole genome. By bottlenecking through a single individual, we've shut down natural selection. Unless it's a lethal, which doesn't matter in evolution anyhow. We collect all the mutations. Let me just leave it at that. We know average rates of nucleotide change at single bases, average across the genome in a lot of organisms. And when you plot that out for different organisms, and you plot it as a function of NE, you get an inverse relationship. So things with bigger NE have higher mutation rates. So we humans have a rate of around 10 to the minus 8 per nucleotide site. You were all born with something like 50 to 100 mutations new mutations your parents gave you that they didn't have. And then down here, microbes there's some things that get as low as 10 to the minus 11. So most newborn bacteria don't have any mutations. Maybe 1 to 3 in 1,000 newborn bacterial cells will have a single mutation. So there's 3 orders of magnitude range of variation. And we think that the best explanation of this pattern is what I call the drift barrier. What's going on as we move so if this is replication fidelity moving to the right, as NE gets larger and larger and larger, you're able to push this distribution to the right. And we don't have much room for improvement. I mean, 1 error every 100 bases that's replicated, that's pretty good. Even 10 to the minus 8 is pretty good, right, an error rate. But the idea here is that microbes are over here with respect to the mutation rate, and then we're more over here because drift is a more prevailing force. And I could go through some theory that tells us what the strength of selection on the mutation rate will be, but I think I'll skip that, and I'll just end with one more example of a very, very different type, and that is based on a lot of work we've been doing lately on another trait. So, as I said, we're interested in patterns of change in mean phenotypes of traits across the tree of life. There aren't that many traits that you can measure in every organism, right? But DNA replication fidelity is certainly one of them. We're doing some work on transcription error rates. Turns out we make huge errors, rates at well, what you might not know if you're not a biologist is, unfortunately, it's convoluted path to going from the DNA to make a protein. We first have to make messenger RNAs out of RNA. And there's a lot of errors that are made inside cells going from DNA to RNA. It's about 10 to the minus 5. So orders of magnitude higher than this. That's consistent with this thing I call the drift barrier hypothesis, because those errors are transient. It's there, transcripts last about an hour, and they're gone. So there's no reinforcing factor. But the other trait that we've been looking at is something everybody has to do. Everybody has to grow, right? Everybody has a growth rate. And if you think about the growth rate, and we'll call this the maximum growth rate, as a function of mass. This is about 20, we have data over 20 orders of magnitude, going from bacteria up to invertebrates. By maximum growth rate, what I'm talking about here is the maximum possible growth rate at any stage in the life cycle. So if you look at an animal or a land plant, usually that's occurring early in development, and then it slows down as we get bigger and bigger. In microbes, it's just the cell division rate. It's log 2 divided by the doubling time. The main thing I want to point out here is that how this pattern falls out. If you look at the pattern in bacteria, it's actually a positive slope. So bigger bacteria have higher cell division rates. And right around here, I mean, there is some overlap, but if you look in eukaryotic cells, it doesn't matter what group, you could look at ciliated protozoans, dinoflagellates, crustaceans, annelids, mollusks, you name it. We have a slope of about minus 0.2. So the bigger we get as eukaryotes, the lower the maximum rate, possible rate of biomass production. Some of you have probably seen papers, by physicists, some not too far from here, a couple states east of here, talking about scaling relationships of metabolic rates and growth rates and so on in organisms. And there's huge debates on whether the regression coefficient is a power of 1/3. That starts to come in when you start thinking about surface to volume ratios and things. Or is it a power of 1/4? There's this theory in fractal sort of dispersion of things. And there's a huge debate as whether the slope is 0.25 or 0.13. And most papers say it's 0.27 or 0.28. That's not necessarily good. Well, at least for maximum growth rate, it's not either one. And you can reject both the minus 0.25 and the minus 1/3.
Is that slope related to that slope?
I don't know if it's just by chance. Yeah, we're aware of they're both being about 0.2. I don't know. I'd be interested if people have any ideas on this. It might just be coincidence.
Maybe all the numbers you get are between 0.1 and 0.3. Is everything you do there?
Well, no, this is a slope of -1. This is a slope of -1. But this has got, it's different stuff on the x-axis. I don't know. It's intriguing, but I guess I'm not ready to come to a conclusion on that. But the point is it's negative, which is expected under the drift barrier hypothesis. The idea is you get bigger and bigger and bigger, and you're getting more and more loaded with mildly deleterious mutations that cost you energetically. One of the main ones is just our genomes are just bloated, right? The human genome's like 1.5% coding DNA, and all the rest is transposable elements and everything else. And we have to replicate that each generation. We're just saddled with that. Microbial genomes are incredibly streamlined. They're 97%, in almost all cases, 97% coding DNA with very little intergenic DNA. And I think this theory helps explain the streamlining of bacterial genomes. You can make this argument just based on this thing I was pointing out before, the cost of, somewhere I was talking about the cost of adding one nucleotide. And as I said, bacteria can feel one nucleotide addition. It all scales with cell volume, and we have much bigger cells. So if you put a 10 kb sequence added to one of your genomes, we did CRISPR on somebody and added a 10 kb stretch, and then measured the change in growth rate due to the additional ATP load, selection wouldn't be able to detect it. So I think this nicely explains genome size evolution. And then finally, what about what's going on here? This is actually the bigger you get, the faster you grow. So it's not even running in the same direction. And an idea here is that we're entertaining is that bacterial NE may be so great that bacteria can feel anything, right? And so they're at a point where drift doesn't matter much at all for long-term phenotypic divergence. At least that's where we are. And one advantage you get in bacteria is the larger you get, what's going on? Well, the larger you get, the surface area to volume ratio is going down, right? And you might think that's bad because you've got less surface area available to pull in nutrients. But it turns out bacterial membranes and cell walls are hugely expensive. So the energy it goes into a bacterial cell to make a new baby, about 1/3 of your entire energy budget is associated with your walls and membranes. And so actually, as you get bigger and bigger and bigger, you're investing less in membrane and walls, and you've got more left over to put into ribosomes and so on. Now you could argue that's the same, that should be true here as well as we get to larger and larger cells, except something else must be going on top of that, and that's why we think this drift barrier starts to play a role.
Is it specifically bacteria that have that upward trend, or is it all little single cell?
No. This ciliates are in here, ciliated protozoans, dinoflagellates are in here. Oh, the other thing I failed to mention, these are all heterotrophs. These are organisms that make their living by eating organic compounds made by photosynthesizers, things like us. But you can also do this thing for phototrophs, which would include the cyanobacteria, all the plankton, the phytoplankton that are photosynthesizing out in the ocean, and including herbaceous land plants, we have data. They also go negative, but here the slope is only about -0.1. Don't get too excited. It's shallower, and we don't know why that is at this point. The data are pretty noisy for land plants and green algae, but the main point is it's negative, and it's certainly not a -0.25 or a -0.33. So this is some new stuff that I think will be coming out pretty soon. So I've tried to give you an overview of the kind of ways we try to think about evolution from a population genetics point of view. My view is population genetics, quantitative genetics is it's one of the great mathematical engines of biology, right? That and biophysics, I guess, maybe biochemistry, but I would say it's every bit as rigorous as the field of biophysics is. And if you want to, my feeling has always been, if you want to entertain a new idea in evolution, it really ought to be, we've been doing this for over a century, starting in, 1918 in Fisher's famous paper, which also started the field of statistics. If you want to introduce a new, fundamentally new idea in evolutionary biology, say we need a new evolutionary synthesis, then I think it needs to be compatible with basic population genetic principles. So one could have a whole litany of courses on population genetics to get into the weeds and the details, but I've tried to tie this back to the two opposing approaches we're taking in our own summer program, and then tie it to some broader data. Most of the program seems to be focused on, how does this plant or this Drosophila or, this vertebrate change, from one environment to another. That's what most of us do in evolutionary biology. But now, inspired by this past work on cell biology that was sort of spawned here many, many years ago, I've gotten more and more interested in these patterns that can be looked at across the whole tree of life. I'm not sure that this kind of pattern at all, if you just took fruit flies, you'd be able to see anything. And the reason it's associated is that within a narrow group, there's a lot of spillage, a lot of variation, even within the narrow group. So you really need to get out to comparisons of organisms with at least a couple of orders of magnitude different in NE, I think, to see these kinds of patterns.
Could you put error bars on either the slope or on the graph?
Yeah, roughly. Let's see, this one is probably about plus or minus 0.1 standard error. This one is probably plus, this is the recombination rate. This is probably plus or minus, I don't know. We'll do the order of magnitude thing. 0.1. That one's really solid. This one, I don't remember, but I guarantee you it's less than, 2 standard errors is less than 0.5 because we can reject the 0.25 model. These aren't the greatest data, right? Because you're dependent on, there's 1,000 data points in there, but, they're all gathered by different labs working on different organisms, and you're hopeful that everybody knows their organism and knows how to grow it at maximum growth rate. A lot of them are based on multiple estimates, so are pretty good. But Yeah, this is noisier for sure than the mutation rate data.
Well, thank you very much.
Okay.
Do you have any more questions?
Well, I have one. It's a little bit silly. One of the most fun concepts in physics is these very robust patterns with all solitons. And you can see them in statistical systems and stuff. Do you have soliton-like behavior in these things? Do you know? Because this could lead to a very silly organism that survives even if it's bad for evolution.
Okay, I got something to learn here. I don't know what a soliton is. I'm sorry. Maybe you can explain it to me. Maybe we have solitons. I don't know.
Yeah, well, for example, if you think of these plus and no plus there,
Yeah.
you'll see them forming some patterns.
Oh, yeah, yeah.
And maybe these patterns are very robust with these fluctuations you have, and some patterns just is really robust if something like this exists.
Well, yeah, I mean, we've reduced it to a pattern. You know, there's a mean and a variance of the number of pluses. And you know, consistent with that number, you can have a dispersion like that. When I talk to physicists, they often tell me, the math here, by the way, is really hard. If anyone wants to help us out, I'd be delighted, because we've spent a lot of time trying to figure some of this out, and it's been, even the simplest cases is bewildering. And I forgot to tell you something else, that it's not all pluses and zeros. So there's big balls and little balls. There are sites with big effects and sites with smaller effect. And that only spreads this thing out even further. So everything I said was probably a conservative statement. But you know, when you have effects that are really small, now nothing happens until you get to be a really large population, because you can't feel them all at all. People keep telling me this theory, you know, the way that it looks is something like, what do you call it? Spin glass models? I don't know.
Another question?
So, if the population size is very large, it can be sensitive to much smaller advantages.
Mm-hmm.
But does it also play in how many generations it takes to spread that advantage to a much larger population?
Yeah, yeah, we have some math that it gives us what's called the time to fixation. It's a little bit complicated, but somebody in the room could help me out. I think the time is roughly, and I emphasize roughly, it's roughly inversely proportional to the selection coefficient, right, to S. The stronger S is, the faster you zoom to fixation. You can almost think about, for a deleterious mutation, as S is the persistence time of a deleterious mutation. It comes in, it takes 1 over S generations to work its way out.
Any more questions?
Well, thank you very much. I enjoyed hearing from you guys in physics, so thanks.